Please start.
Well, very good afternoon to everyone, especially to our guest, Dr. Chandradesh Ghosh. So we welcome to the third session of this SDP, Student Development Program online. This is the webinar series we have initiated several months ago for the large benefit of our student. And today, as you know, our guest speaker is Dr. Chandradesh Ghosh. Chandradesh Ghosh is a postdoctoral researcher at the Max Planck Institute of Colloids and Interface, Department of Biomolecular System, Mühlenberg, Potsdam, Germany. It is a suburb of Berlin. So Dr. Ghosh will address today's session. So prior going to invite Dr. Ghosh, I would like to introduce you a bit about Dr. Ghosh. He has performed his doctoral degree at Jawaharlal Nehru Center for Advanced Scientific Research at Bangalore under the supervision of Professor Jayant Haldar. After first earning the Master in Chemistry from the University of Delhi, he did a doctoral work on membrane-active small molecules against drug resistance pathogens that has led to an anti-Ebola drug candidate. Dr. Ghosh has contributed to the sum of knowledge in the field of the publication of several highly cited reviews. He has won several awards, including the prestigious Gandhian Young Technological Innovation Award in 2015. Since 2017, he has been working at the Max Planck Institute for Colloids and Interface in the group of Professor Peter H. Zeeberger. His current research work focuses on targeting neglected tropical disease. He is the recipient of the 2020 RSC Medicinal Chemistry Emerging Investigators Leadership Award by the Royal Society of Chemistry, United Kingdom. This prestigious lectureship is awarded to those researchers who have made a significant contribution to medicinal chemistry in their earlier career. So, dear colleagues, just a bit about the session. So here in this session, Dr. Ghosh will focus on infectious disease continues to be one of the major contributors to a human morbidity. The rapid rate at which pathogenic microorganisms have developed resistance against frontline antimicrobials have compelled scientists to look for new alternatives. Due to their vast antimicrobial repertories, substantial research effort has been dedicated toward the development of antimicrobial peptides as alternative drugs. So I suppose I should not go in detail, let it be a top secret, and let Dr. Ghosh to reveal the factual information. So now I would like to invite Dr. Ghosh for conducting this session. And prior to that, as usually, I requested all of you to mute your mic and camera. And if you have any query, you can place in a chat box. We have a dedicated faculty members led by Dr. Renu Singh, Dr. Sonika Sethi, Dr. Soumita Talukedar, and they will record your question on your behalf. And after the session, we will open the session for a question answer, and our dedicated faculty will ask a question on your behalf to Dr. Ghosh, and I'm sure Dr. Ghosh will try his level best to satisfy with his answer. So once again, thank you, Dr. Ghosh, for your time and conducting this session. We are much happy to have you here. So I may kindly request you to initiate this session. Thank you, please.
Thank you, Dr. Thakur. Special thanks to my old friend Soumita Talukdar and also Sonika for inviting me. And I'm very glad to speak to young, bright minds who will probably be leading the country in a day's time. And with that, I will start. The title of my talk is The Story of Aryl-Alkyl-Lysines. and how one class of molecules can be used for treating different varieties of infectious diseases. So we know of infectious diseases. Right now we are undergoing a pandemic. But we should be aware in a little detail, around 57 million people die each year for infectious diseases, and around 25% of those deaths are caused by infectious diseases. Sorry, I misquoted it. Around 15 million people die of it. And what are infectious diseases? They are caused by different pathogens, right? So you've heard of bacteria, you've heard of fungi, you've heard of parasites. Parasites are the ones that cause malaria. And also in India, we have a problem called Kala Jar. Everybody knows about it. It's caused by this parasite called Leishmania. Of course, you've heard of Ebola virus in 2015. And this young fellow, COVID-19, has already taken 1 million lives, actually more than that. And everybody is suffering. And maybe if it wasn't for COVID-19, I would be speaking to you in person, which of course I would like to do. So I've been told that I'm addressing a crowd of both bachelors, masters, and from different backgrounds, and as well as esteemed faculty members of different backgrounds. So what I will do is I will introduce a bit of chemistry and a bit of microbiology. I will try to speak in the terms that I was learning when I was a student. And if you have any questions, please feel free to ask me, whatever be it. And all that I want from this presentation is that at least one or two of you may remember at least 5 to 10% of what I say, and then maybe that inspires you to do something for India and for the rest of the world as well. Right. So these microorganisms are the causative agents for infectious diseases, and they can have different manifestations. What do I mean by that? That they can survive in different forms. Some of them can be free floating, as you can see here. These are called planktonic cells. This is represented by this image here. So these are bacteria that are free by themselves. They can also form this kind of clusters. These are called biofilms. These are called microcommunities, and inside this matrix, the biofilms of the bacteria are surviving within. And some of them can also go inside your immune cells. Some of you might have heard of immunological response to COVID-19, and people are exploiting it. That is, our body's immune system is functioning. And for that, you need certain cells, like, for example, macrophages. You've heard of these, maybe. And some of these pathogens can actually go inside your immune cells and survive within. So that makes it even more difficult to treat, because the same cells which are supposed to kill it are now being a host for this, for these kind of pathogens. And these are important problems that everybody needs to be aware of. When you go to the doctor as a sick person and the doctor prescribes antibiotic or antifungal agent, whatever it is, you have to understand all of these to really understand the importance of dosage. Anyway, we understand that there is a big problem. And the problem is, you've heard that the doctors say that, well, I don't have an antibiotic right now because the bacteria or the pathogen is getting resistant to it. What does that mean? That means that the concentration at which the compound was acting before is not relevant anymore, because the bacteria have developed a resistance. Or not only bacteria, it is in case of parasites, fungi, viruses, even cancer. You've heard that the drugs are not effective. And this is a problem in world health right now. And this is called antimicrobial resistance. And it is projected that if we don't take care of it right now, it's going to cost around 10 million deaths by 2050. And it's going to be extremely difficult for healthcare systems all around the world. And for a developing country like ours in India, it's a massive problem. And we need young minds like you to sort of understand this problem and contribute in that direction. Now, who's supposed to take care of this? There are drug agencies, like you heard of the Drug Controller General of India. They are the ones who allow new drugs to be approved in India. Here I'm quoting the FDA. This is the American agency, American Drug Agency, which approves new drugs. And you can see that from 95 to 99, there were around 33 drugs approved, and it slowly decreased. Right, this bar is actually representative of viruses. The black one is for bacteria. You can see that till 2009 there was a huge decrease in there was no new antibiotics being approved. And then the world understood the problem of antimicrobial resistance. That just means, like, let's say you're playing cricket or football or whatever, and then you get a cut. And if from that cut, if there's any pathogen which goes inside your body, you might be able to die from it because there would be no antibiotic or any drug left to treat you. And because of this antimicrobial resistance. And so people started working on it, and then the FDA started approving more drugs. There has been a better state right now, but that doesn't mean that we have to continuously produce more drugs and hopefully be able to defeat the bad pathogens. So now I come to this point, like, okay, I understand that there is a need to discover new drugs, but how do you find it? Where do you find it? You've heard of Alexander Fleming. He was the first person who discovered penicillin, which is the first antibiotic that was used. But he has done many other things. So he discovered these things called antimicrobial peptides. You know, I will tell you this particular story. I don't know whether you've noticed. You see, when dogs, they are like, they have a wound, you will see them lick the wound, right? That is because they release proteins called lysozyme, for example, which has antimicrobial properties. That means anti is something against, and microbe is like the pathogen that is killing it. So to have that, lysozyme is something that is present in all of our saliva, is actually an antimicrobial peptide. Look at plants, look at other animals who do not have a healthcare facility like we do. They don't have a doctor. How do they survive? I mean, they're also infected by bacteria, right? Or other pathogens for that matter. That is because their body produces this thing called host defense peptide, which can tweak your immune system to make it work better or kill the pathogens directly. And these are found everywhere. It's on your skin, in your eyes. Like, the eyes are most exposed part of your body, but they don't get infected. So this, because of the presence of this host defense peptides. And chemically speaking, they are around 12 to 50 amino acids long. Everybody probably knows of amino acids. And they're a mixture of cationic and hydrophobic amino acids. So by cationic, I mean that have positive charges, and hydrophobic is something that repels water, that likes oil more. So they have these two regions. One is cationic and one is hydrophobic. And then the bacterial cell membrane is negatively charged. So you can understand, if this is positively charged and this is negatively charged, they get attracted. And then through the hydrophobic domain, the water repelling domain, they interact with the cell membrane and cause holes in it. Okay, and then that is how they kill it. And these antimicrobial peptides actually possess antibacterial. That means they can kill bacteria and their biofilms. Antivirals, which is very important right now, like kill viruses. Antifungal, against fungal pathogens. You've seen some fungal pathogens, for example, on toenails, you'll see, or the rings that you see. Those are caused by fungi. So these are also important. And parasitic, that means killing malaria or kala-azar, cancer, everybody knows, and also have immune modulatory properties. That means they tweak your immune system. However, these cannot be readily translated into the market because they are not very stable. The moment you put them, in our body, we have these things called proteases, which break down these proteins. And they're toxic. That means they cannot distinguish between the good cells of your body and the harmful pathogens. And they're expensive. So for a clinical setting like India, it's a very big problem because not all of us can afford a $100 drug, or it's very expensive, 1,000 rupees. It's not a matter of joke. There was some peptide antibiotics. That means these are, you can see these are peptide bonds, that CO and NH, and these are various amino acids put together. So these are peptide antibiotics. One is called Daptomycin and another is called collistin. So collistin was used by FDA. It was approved by FDA in 2007, but bacteria grew resistant to it in 2013. Same with daptomycin, it got approved in 2003, and resistant development was reported in 2005. So that means these guys do not even survive in the clinic. The moment you start applying them, they get resistant. And why do you think that happens? Because, let's say you go to a doctor and you have a sore throat or whatever, and the doctor tells you that, well, you have a bacterial infection, tells you a bacterial infection, and then prescribes you antibiotic. And he says you like, well, take it twice a day for at least 5 days. On the third day, you start feeling better and you say like, okay, I'm fine, so I don't need to use it anymore. But that is where the problem lies. you should always complete the course. Why? Because as you take the antibiotic, the entire bacterial spectrum is killed. But if there is little bit of bacteria that is still remaining, they are getting very little of the compound. They start developing resistance to it slowly. So we are the contributors to this particular problem. Especially it is rampant in India because we all have a habit, chalo theek hai, tabiyat theek hai abhi, so we stop taking the medicines. And this is something that I want to educate everyone listening to me right now. Whether you take an antibiotic or an antifungal or an antiviral, it is extremely important to complete the dose, what the doctor prescribes to you. Otherwise, you would be responsible for the death of your near and dear ones. You don't even know how, but this is how it is contributing. So please take care of this. Anyway, so there are peptide antibiotics present, but as I said, that they have problems of stability, toxicity, and cost. So how do you solve this problem, right? You can understand that there are these three problems, and these need to be solved. So scientists all around the world started making mimics of this. Mimics means something which emulate the properties of these compounds, but not structurally similar. So there are many things that have been done. For example, some of them have used polymers. So and oligomers. This may be don't get bogged by the structures. Okay, these are if you understand it, like if you look at it, if you understand it, you will remember it. For example, amino group here represents a positive charge. Anything that has NH2 can take up one hydrogen and become positively charged. And whenever you see alkyne chains or alkene chains, you see these are hydrophobic. So they have hydrophobicity on one side and cationic positive charge on the other, right? You can see this everywhere. You see there's another positive charge here, another positive charge here. And there is this hydrophobic part here as well. All of these molecules would have the same. You see, this is another positive charge, another positive charge, and CF3 is a very hydrophobic. Aromatic rings are very hydrophobic. So they play between this hydrophobicity and cationic charges, and they mimic the properties of antimicrobial peptides. So when I started my PhD, all of these molecules, not all of these were known, right? So people were started working on this, and my supervisor came and told me, why don't you try to work on this direction? And then I was spending a lot of time trying to think how to do it. And after reading some of the earlier papers, some of the earlier scientific work that was done, I realized that you need to have at least 2 positive charges. I mean, that means this amine groups, and you need to have at least 2 hydrophobic bulky groups. That means, like, for example, the aromatic rings or even the lipid tails like this you see here. This is an alkene present in a lot of alkanes rather. And so you need to have this kind of a lipid tail as well. And you need to have very cheap starting materials. Otherwise, it will be a problem for countries like India. And for having cheap starting materials and to make the price very low, you need to have minimal synthetic steps. So then I thought that what are these? Like what is the easiest molecule that I can find that will have 2 positive charges? Now you might be doing in your classes the different structures of amino acids, and you might have heard of arginine, histidine, and lysine. You see, there's a COOH group here which is negatively charged and an NH2 group which is positively charged. So overall, this is neutralized. However, in their side chains, when they have this another additional positive charge, that means you have at least 1 positive charge. If you do something here to the COOH group, if you functionalize it, that means you will have 2 positive charges. So that is, I already fulfill the criteria of having 2 positive charges. So this is what I thought, that I will work on this kind of molecules and I will make a modification here. What kind of modification would I do? So that I can introduce hydrophobic bulky groups, right? And I also have to keep in mind that these are cheap starting materials. So this is exactly what I do. Keeping these in mind, I think of L-lysine. As I mentioned, there are 2 positive charges, and if I functionalize here, I can do it. And what kind of hydrophobics? I thought the basic thing is like long alkyl tails, and then I can use hydrophobic moieties like the aromatic rings. So some of you might know that amines react with COOH groups to form amides. Those are the basis of all the peptides that we have. So if I introduce something like this here, that means I introduce hydrophobicity and the 2 positive charges remain. right? So I started with the very simple molecules, right? So these are called alkyl amines, and these are different lengths: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There's 12 carbons, and there is an amine here. So with that amine, I'm going to, conjugate to the carboxylic acids. And this is how we do synthesis in the lab. So to make these molecules, I first had to take alkyl bromide. So imagine I have a Br here, and then I put sodium azide, which basically makes it the N3 here. The bromine is replaced by N3. And then we reduce this N3 to amines, to these. And then once we have these, I couple it to the lysine. This is the Bock protected lysine. So, like in synthetic chemistry, we need to understand that to do reaction in one center, we have to close down the other. And so the amine groups are protected by these groups called Bock. And then you can do this reaction in presence of HBTU, DIPA. So these are reagents. You don't have to remember them. As you grow older, you will do it anyway. And then you remove the protecting groups and you get the final compounds, right? And then we checked at its antibacterial activity and hemolysis. So I will tell you what I mean by them. So by antibacterial activity, I need to know whether these compounds can kill bacteria. So essentially what I'm trying to do is I'm trying to understand at what concentration I'm going to kill the bacteria. So there is a minimum parameter that is known in this field, like this concentration, like in micrograms per ml, around 5 micrograms per ml is okay, is acceptable. So that means if you treat a bacteria of 10 to the power 5 cells with 5 micrograms per ml of compound and all the bacteria are killed, that compound is good rather. And how do you determine that? So you determine it using a microtite. It's called a 96 well plate, and I will tell you briefly how the experiment is done, right? So now first you grow bacteria. The microbiology majors probably would know what I mean by growing bacteria, and of course some of the professors here know. So you grow it to a certain extent. So they usually go to 10 to the power 9 cells, and then you dilute it to 10 to the power 5 cells per milliliter. So that means 1 milliliter of solution will contain around 10 to the power 5 cells. And you treat the compounds at different concentrations. So what you do is, in this particular well, you start with the highest concentration on this well. This is like a very small microtiter well, it's called. You can put in 200 microliters of a solution, right? So you start having the highest concentration on this well, like for example, let's say 100 micrograms per ml, and then you dilute it 2 times. So this second well comes to 50 micrograms per ml, and then you dilute it 2 times further. This is 25 micrograms per ml, and you keep on going 2 times dilutions, and until you reach this particular point where you don't add any of the compound. Okay, to that same concentration, now you add bacteria. In all of them, you add the same quantity of bacteria. Now what happens is, if the compound is active, then it remains like this. Okay, there is no growth in bacteria, and it's clear solution. However, if bacteria starts growing, it is not clear anymore. It is turbid. It's almost like milk, or not. It's a little too much, a little lower than that, but it gets very turbid. And the concentration at which you see this demarcation is the minimum inhibitory concentration. That means the concentration at which it can kill the bacteria. So if you start from 100 here, so it's 100, 50, 25, and this is 12.5. So the minimum inhibitory concentration of this is 25 micrograms per ml. So these are 2 different bacteria, E. coli and S. aureus. I'm just giving you this is a general example. So from this experiment, you can conclude that the compound probably kills E. coli at 25 micrograms per ml. And if you go further, 12.5, 6.25, 3.125, and 1.56, and kills S. aureus at 1.56 micrograms per ml. So these are the MIC values that are needed. So the lower the MIC, the better the compound. However, there is another question. At this concentration, does it harm yourself? Does it harm your red blood cells? And for that, you have to determine the hemolytic assay. So if at this concentration, it also kills red blood cells, then you can't use it, right? Because at the same concentration, it is harmful to you. So you have to do imagine in case of in this case I put bacteria. Now I'm going to put red blood cells. If the compound is going to kill the red blood cells, this is how it will look like. This red spots here, 1, 2, 3, 4. If it does not, then the red blood cells remain intact, and it is represented by this. And using the same procedure, you determine the hemolytic concentration as well. So the compounds that I had made here in this particular step, with all of these C12, C14, C16, C18, C20, I saw that they were, so this is represented by S. aureus and E. coli, these are the two bacteria. They were killing at around 20 micrograms per ml. And the hemolytic concentration is 120 micrograms per ml. So that means you can see there is a huge difference, 6 times difference. So at 20 micrograms per ml, it wasn't killing the red blood cells. So that means this compound is doing better. So as I said, 20 micrograms per ml is not a very good number. You need to go even lower to 5 micrograms per ml. And you can see that C16 here, like this particular compound, when you conjugate to the lysine, you get this particular value. And the hemolytic concentration is lower. That means this compound is a little more toxic than C12, but also a little more active, that it kills bacteria. Also, it's a little toxic at 80 micrograms per ml. So this is the basis on which I'm going to talk about all the further studies, right? Then I thought, okay, if the single chain is like that, maybe I will introduce 2 chains, because then they would be able to interact much better. So I made these amines. You can see on 2 sides there are 2 lipid tails, and I followed the same procedure, and I determined the antibacterial property. And you can see that this particular compound is like really, really low. The concentration at which it kills S. aureus and E. coli, those are the harmful cells. However, the red blood cells, it's very high. It's around 100. So that means this is a very, very good compound. And here we also determine how fast does it kill the bacteria. So you add the bacteria at 10 to the power 5 cells and everything, and you can see that they kill within an hour, within half an hour actually. So these are very fast at killing. Now we determine the mechanism of action. That means how do these compounds act on the bacteria. So for that, I have to introduce 2 different things. So one is the gram-positive bacteria and gram-negative bacteria. So this is determined by what staining they take up. The microbiology honors people would probably know about this. So this is a peptidoglycan layer, and this is the plasma membrane, and the compound is supposed to infiltrate both of them. So these membranes are there. The plasma membrane is present in human cells, but it's different in bacteria. So first we see if the membrane is depolarized. What does that mean? That means that there is a potential gradient between the 2 sides of the membrane, on this side, on the outside. And if you depolarize it, if the potential gradient is not maintained, then the bacteria loses its integrity. And this is determined by this depolarization assay. You don't need to know the details, but it's done using a dye, which is basically a fluorescent molecule. So this dye starts being fluorescent. That means it's increased in fluorescent. You can see, I mean, it shines at a higher wavelength. You can see that sudden increase once you add the compound to the bacteria. So that means the membrane have been depolarized. Similarly, you can also see whether the inner cell membrane is also infiltrated. That means there are pores inside. This particular N-phenyl-naphthylamine will go on to the outer membrane of the bacteria and sit there. Once you damage the outer membrane, the fluorescence increases, and you can see it in this particular graph. So once you add, these compounds can act by membrane depolarization, outer membrane permeability, and also inner membrane. So this membrane is also, if it is damaged, then this dye fluoresces. So it was concluded that the mechanism of action of these kind of compounds is through that. And then, as you might know, that finally to prove a concept, we have to do it in mice models. So here I had taken a murine model of burn infection. That means, imagine if you have a burn, and in that burn you have been given bacteria, or like there's an infection of that burn. That means the bacteria is growing, and sometimes you've heard that people have to amputate their arms, or like they have to cut it off because it should not spread to the other parts of the body. So we did that in mice, and this is called the burn infection model in murines. And if you put 10 to the power 9 bacteria, when you treat it with the compound, it is brought down by 2 logs. That means 2 10 to the power 7 cells, which is already very good. There is a classical compound that is used currently. It's called collistin. I had introduced it. That kills around 10 to the power 3 cells. So from 10 to the power 9 cells, it brings down to 10 to the power 3 cells. That's a huge difference. So that means this antibiotic is very, very good. The compound that I made was good, but not as good as this. The lower this is, the better it is. And then I wanted to show that the bacteria do not develop resistance. So that means that as you might have heard from the doctors, like, okay, because of resistance, this antibiotic does not work. I wanted to see if my compounds will develop that resistance. But turns out, I'm not going to go into the details of this particular experiment. The thing is, these kind of compounds do not get resistance easily. Okay. But then I wanted to make even better compounds because there was a little toxicity, as I had said. So I had made various different molecules using hydrophobicity, as you can see here. And here in this particular case, this was my best compound, and I used the naphthyl ring here and the hydrophobic alkyl chains and connected to lysines to create this sense of compounds called NCK series. This is very simple, as I said. So you've heard in chemistry, the chemistry graduates might be able to know this. So this is a Schiff-based reaction. So when you put an amine against an aldehyde, it forms the imine, and the imine needs to be reduced by a reducing agent, sodium borohydride, and then you get reduced, and then you put a salt, like you put HCl and forms this salt. These now you connect to the lysine groups, like previously I had shown, using that same reagents, and you have this compound, and then you deprotect these groups. You have two NH2 groups contributing the two positive charges, and the two hydrophobic moieties are there. So these kind of compounds were also checked for their antibacterial activity. So if you have an 8-octyl chain here, it's a NCK8. If you have a decyl chain, that means C10, it's NCK10. Vancomycin and tetracycline are currently used antibiotics. So we checked bacteria which are resistant to these drugs, and we found that both of these compounds are acting much, much better than these compounds, right? And we also found that these compounds were not very toxic in our red blood cells as well as mice. So it is safe for use. And then we went about checking their activity. As I said, they are killing very fast as well, like the previous ones. And they are also not developing, the bacteria are not developing resistance to these. Now, as I had introduced before, that sometimes these bacteria form colonies called biofilms. Okay, these are extremely difficult to get rid of. But these compounds were able to kill it. So when you have this cluster, this is the thickness. This is a microscopic image of bacteria inside biofilms. Okay, this is what it looks like. This is the thickness. So when this is negative control, that means there is no compound added. You can see that this is very thick, and the bacteria is all over. These green dots are bacteria actually. But when you treat it with NCK10, this compound, this thickness decreases, and you can hardly see any bacteria. Of course, some remain, but almost 100%, 90% of them are completely killed. So that means these compounds have antibiofilm activity as well. And these are some of the cells that are present within the biofilms. This is not very important, but they can also have activity within the biofilms. And as I showed before, these compounds also work by depolarizing the cell membrane of pathogens and by infiltrating the membranes. How does it look once you study it under the microscope? You can see here, this is an SEM image. This is scanning electron microscopy image. So these are intact cells. So these are not given any kind of compound. The moment I give the compound, this is what it looks like. You can see these cells, they have holes inside, and it's completely ruptured. So that means the cell membranes are completely damaged upon use of these compounds. Again, we had checked in mice models, and we had taken two very important bacteria, Pseudomonas aeruginosa, which causes lung infections a lot. So in ICU patients, actually, this bacteria causes a lot of problems. And Acinetobacter baumannii is also a rising problem all over the world, especially in India. So as I said, collistin is a currently used antibiotic. It is not as good. My compound is not as good, but it's already very, very good against this kind of infections. However, we have to keep in mind that the bacteria get resistant to collistin very, very easily, but not to my compound. So that's where it makes my compound much better. And these are extremely expensive and toxic, and my compounds that way are not toxic at all and are not at very, very cheap. So with that, I moved on. So with this particular set of compounds, I have done other tests against other pathogens. For example, fungi, as I said, this is what fungal look like. Unfortunately, it's not very nice to look at, but this is a fungal infection. This is what it looks like. And you need to use it. You'll be given creams. You've heard of fluconazole. Sometimes they use this compound called amphotericin B. And this is my compound. Again, you can see the panel of MIC here. I will say that the lower it is, the better it is. Amphotericin B, you can see, is as active as my compound. So these are my compounds are doing very well against the fungal cells. And this is again the time of killing. You can see it kills very fast. So the lower the number of these cells, that means the pathogens are killed. And this is untreated. When you don't add any compound, it keeps growing. Here, it's gone. It works against the biofilms of fungi as well, as I had introduced before. But fungi also form biofilms like bacteria. And they again work by damaging the cell membranes of the fungal cells. This can be observed even microscopically. Basically, essentially the same experiment. And then against Ebola virus. So you've heard of Ebola virus. It was a huge problem in Africa. And one of my compounds was selected by Public Health England. They were an organization who were conducting anti-Ebola studies, and they came to know about my work, and they contacted us and said, like, can you give us your best compound to check? And they had taken many compounds and selected 60 compounds for final studies to have anti-Ebola activity. And this NCK8, one of my compounds, actually did quite well. It was the best that they had studied amongst all of these compounds. And that's why, I mean, we got a little highlight from the media. Times of India covered us, the Hindu covered us at that particular point of time. And so this compound was doing well. And in fact, when you put it in animals which have Ebola infection, at least there was some survival observed. And at that time, this was creating a lot of noise. And anti-malarial activity. So for that, I need to tell you a little bit about the Plasmodium falciparum. Nobody needs to be introduced to malaria. At least there are 228 million cases of malaria worldwide, even 2 years ago. And around many people die. You know, you must be having one or two relatives who've probably had malaria, or maybe some of you have had malaria. So it's caused by this agent called Plasmodium falciparum. So once the mosquito bites you, it releases this particular parasite. This goes into your bloodstream and goes into the liver cells. And inside the liver cells, they start dividing. And then they burst open the liver cells. That's why your liver starts failing. And then it go into the bloodstream. In the bloodstream, you have heard of red blood cells. They infect the red blood cells, and then they grow inside and then burst open the red blood cells. And then they divide within, and some of them are taken up by the next mosquito from the blood. And then inside the mosquito, there is another cycle that goes on. So when you have this cycle in the red blood cells, that is when you start feeling unwell. You have the problems of parasites. One of my compounds actually was doing very well at 1 micromolar. NCK6 was able to kill the parasites. And these are some of the images that you can see. This is what it looks like under the microscope. This particular stage, these two, you can see here. These are called schizons. This is the schizon form. And initially it's called a ring. And this is what a ring looks like. So we could see that the compound was stopping at this particular step and not allowing this to happen. And we also saw that they studied the mechanism of action. They were perturbing with the membrane potential of the parasites. And even mice which were given this particular compound did not die because of, their survival was prolonged by at least 5 days when you are given this particular compound. And with that, I flew from Bangalore and I moved to Potsdam. And I started working in one of the best departments in the world for this thing called carbohydrates, which are things that you've heard of. But I basically worked on sugar-based gold nanoparticles, and I have conjugated different drugs and different other sugars to have immunomodulatory properties, and also proteins for having autoimmune diseases to solve these kind of problems. And these are some of my ongoing projects. I'm not going to talk about them today, and maybe next year if I get another invitation to talk to you guys. But by that time, I hope that at least 1 or 2 of you would be starting a career in science and making changes to Indian healthcare system and making India much better at all of solving these problems. And with that, I thank all the people that I've worked with. As you can see that I have a lot of collaborators from all over the world, including within India, in the US, in France. And here I have a very nice group of people who are from all over the world as well. And all of my co-workers in India are doing very well. Professor Haldar is a famous scientist from India right now. And I would be happy to connect with you guys. And this is a picture from one of my friends' place in Berlin. This is what Berlin looks like. You can feel free to reach out to me. And I thank especially my old friends, Shomita and Sonika, and of course, Dr. Thakur for introducing me. And I thank you for your attention.
Well, thank you. Thank you, Dr. Ghosh, for such a nice talk, discussion. And I'm sure it would be much fruitful, beneficial for not only students, but also for the faculty members working in this field. And now we open for the discussion. So, Dr. Renu Singh.
Yes, sir. Thank you, sir.
Yeah.
Thank you, Dr. Ghosh. Sure, sir. We also hope that some of our students must have been motivated with your such a motivational talk, such a scientific, and I should say that it is a motivation because most of your work is your original work that that must have motivated our young budding students. So here are the few queries from our participants and the faculty side. So, sir, I would like to put their questions on behalf of you so that you can clarify those questions. Here is our faculty member Shashank Shivaji Kamle, sir. I would like to request Shashank, sir, please unmute yourself and ask your question. Hello. Shashank,
Yeah,
sir, are you
actually,
there?
I got disconnected. Can you hear me?
Yes, sir. Please put your question before our guest, sir.
Actually, my connection got disconnected high. So can you please read the questions? Actually, I can't see my questions.
Yes. Sure, sir. Dr. Ghosh, first question was, do you also check a tissue-specific toxicity of these drugs in terms of inflammatory marker rather than taking a cell death as an end marker?
So we have tested against various cell lines, like for example, HeLa cells. These are human cancerous cells, and also macrophages. And we don't have any intermittent markers because, in my case, it's not entirely relevant because I would like to see whether the cells survive in the end or not. So I look at the endpoint as like the death of the cells rather than having a marker in the midway.
My question to you is that, can I interrupt? Yeah. So toxicity, toxicity can also be in terms of not, you can't, the death is the end point of toxicity. Toxicity can also be in terms of inflammatory inflammation. But the cell is not
absolutely.
surviving.
Yeah, absolutely. So I completely agree with you. But in these kind of particular compounds case, because we are looking at the problems that happen directly, right? So like whether the cell membranes are damaged, and then you see that. I mean, I completely agree with you that these are another thing that might have to look at if you want to have a clinical trial of this particular compound, that what it is doing downstream of the inflammatory pathway and everything. I have done some immunological studies with these. We have seen that they have some activity on TNF alpha and interleukin 6 and everything. I didn't want to present this because it's hardcore immunology. So we have some idea as to what they do, but for that kind of a study, it is probably better to do it, for a clinical setting. But for my experiments, I did not need to do that. But I completely agree with you that it can have other kind of parallel problems with respect to toxicity.
Thank you.
Renu ma'am, can you please read the next question?
Yes, sir. Yes, sir. Sir, the second question is again from Shashank, sir. Why there is a slow development of drug resistance to your compounds? Any reason involving the structure?
Absolutely. Okay. Since you brought this up, I will describe this to you a little details.
And there's one more slide that when you explain this particular compound on the persister cell in case of biofilms.
Okay. Since you're aware of it, I will answer it. Okay. Okay. All right. So So I will just, I don't know whether you can see my slides. Can you see my slides now?
No,
Oh,
sir.
It's not visible.
okay. I'm sorry. Yeah. Right. So thank you for asking me this question. Actually, I would have liked to anyway bring this up. So this stems from antimicrobial peptides, right? So what happens is the antimicrobial peptides actually kill by damaging the membranes. Now, for all the antibiotics that are present in the market, they are probably acting on either proteins or on the DNA, or they are targeting physiological functions of the bacteria, right? They were starting, let's say, stopping replication, stopping translation, and something like that. However, these compounds are not doing anything like that. So antimicrobial peptides, and also I've seen that my compounds also have this functions, right? You see, okay, I'll go to the next one here. So these are also acting on the membranes. So the membrane remains irrespective of whether the compound, whether the bacteria is dividing or not. So their physiological functions are down when the bacteria is not dividing, and that's why some of the other antibiotics are not able to, the bacteria develop resistance to some of those compounds. But these are not acting on replication, translation, everything. It is directly going and killing the bacteria. So that means that is why the survival of bacteria is completely there, gone. They cannot develop resistance for this particular reason. However, there's a caveat. It does not mean that they will not grow resistant complete later. What it does is, you can see this, I've done it in 20 passages. In 20 passages, they do not develop resistance. I haven't done beyond 20, which is a very good number. 20 is already a very good number. But if you go like, let's say 50, I would expect this MIC value to probably go a little higher. So, I mean, I'm not saying that it will not get resistant at all, but it's very, very slow. That is because bacteria, the energy that it has to, cross to have this kind of modifications on its cell membrane surface, that this interaction of positive and negative charges do not happen, is extremely difficult. And you asked me about the persister cells. So as you know, the persister cells are there because after treating with an antibiotic, some of them remain because they shut down their metabolic activity. That means they're not dividing anymore or growing anymore. However, these compounds are acting on the membrane. So irrespective of whether their mechanism is shut down and everything, membrane is there, and the membrane is lysed. That is why they act, and that is why they survive. Rather, I hope
Thank
I'm
you, sir.
able to answer your question, but please feel free to ask more if you have.
So Sashank sir has another question. Have you checked its efficiency against a microbial infections also?
No, we haven't done that test, but that is a very good, we had thought of it. We had actually thought of having a bacteria and a fungi, or two different species of bacteria. But I at some point you leave PhD, and I left. And
because your answer of developing resistance will be, I think, answered there in case of polymicrobial infections, other than testing individually.
It is irrespective of that, because in every case it is going to damage the membrane. We have seen it in case of all the bacteria. So I've done a test against a panel of gram-positive and gram-negative bacteria, and in every case the cell membrane is.
Have you tried in combination?
Yeah, I've done it in combination as well. I have not taken two different species of bacteria for that matter, but I have done like the compound I've taken in combination with other antibiotics also, actually. But one cannot present all of that. But what happens is, like, if you have the compound present, both gram-positive bacteria and gram-negative bacteria, their membranes are completely lysed, and I have actually proof of that even in the slides actually. So because it is acting like that, we expect, actually one of my juniors, I think, did not with my compounds but some of his compounds, which are based on the same principle, and I think he had good results, but I can't quote you on that. But this is a very good suggestion, that is this probably would have different
the bacteria and the other microbes who are living in polymicrobial infections, they tend to develop resistance faster, at a faster rate than living in individual communities.
Yeah, that is because of horizontal gene transfer and all of that. But then again, these kind of things happen once you have an antibiotic which is working on the physiological processes. So that is why I expect, I haven't done it, so that's why I cannot confirm it to you. But I expect for my compounds, it will not happen like that. But I'm not saying that, it cannot happen also.
Okay. So, Mervin, our one of the student Mervin, he has a question that what is a binding site for cationic end of aryl or alkali amine molecule?
Okay, thank you, dear student, for asking this question, and I don't know if I can, I don't have the access to a board, otherwise I would be able to explain it better. Let's try this way. Hold on. Okay, so the first question is, what is the,
Binding
how
site.
does bind? So there is no binding site, rather. Okay, so you see they damage the membranes, right? So the bacterial cell membrane is made up of phospholipids. So that means there's a phosphate group, that's a negative charge, and the lipid tail, right? So the positive charge of the cations get attracted to the phosphate heads. So it's positive negative interaction, so they come closer. And then the lipid tails are water repelling and hydrophobic molecules. And I showed that in my compounds also they have these, you see these two, these are all hydrophobic. So essentially what they do is, I'm sorry, okay. Maybe this is not a good slide. Actually, I don't have that. So if you look at, okay, this is, maybe you can do it yourself. So this is the hydrophobic moiety, right? And this is the lipid, another lipid part. So these two sites actually go and get positively they're attracted to the negative charge of the phosphate heads. And then from that phosphate head, you know that there are two of these lipid chains which hang. And then this part interacts with them. Actually, we've done computer simulation studies with that. We've never published it, but I have data to show how the membrane is completely damaged because these two hydrophobic parts interact with the lipid part of the compounds, or rather the cell membrane, the phospholipids, and then they're completely damaged. So per se, there is no active site like that. They go and damage the membranes in general.
Uh, yes, sir. Anushka Yadav, she is asking that is it effective against the spores as well as we know that some bacteria under unfavorable conditions form a spores and then germinate again when favorable conditions are available? So,
Very good question. I unfortunately, maybe I have that here. No, I don't have it. Unfortunately, I'm sorry. So we have seen. So I will try to answer the question directly. So since you know about the spores, you might be knowing about another bacteria called Clostridium difficile, right? So Clostridium difficile is one such spore forming bacteria. And we have seen that this particular compound actually does kill the cells as well as the spores, but the concentration at which it kills the spores is much higher. So in principle, it can kill the spores, but at a higher concentration. So I would not say that it's a sporicidal compound that way at the same concentration. But if you increase it, yes, it does kill the spores.
Just I have one last question.
Sure. Sure.
Yeah. This is Shashank. So why one question to you is that you checked for the resistance for 20 cycles, right?
Can you repeat it again, please?
You checked for the resistance for 20 passages, I guess, right? You said 20 passages.
Yes.
20%.
Yes.
That's a very good number. Just want to know whether did you get persister population at each of them and at all the concentrations of the drug?
So again, to get persister population, you have to treat with an antibiotic which targets the physiological cells, right?
Okay.
So this compound is killing all the cells. So it's damaging on the membrane. So you don't have any persisters from this particular compound because everything is killed. You can't see any bacteria. What you probably might be another way to put this forward is like, do you have like for example, if you have, let's say, when you do a frequency of resistance experiment, then you find some of the compounds, some of the bacteria which might develop resistance to it. That I had done one study, but I did not have very good results, like to even have a conclusion. I was having one day I was seeing that it does not have any
I
population.
mean, I just want to say about stochastic resistance, not the long-term resistance.
What I don't know this term. I'm sorry. What is
Stochastic
stochastic?
resistance.
I can you please tell me what this is? I have no idea
Yeah.
about this actually.
So it might not be resistant, but it might survive that particular dose.
Yeah, but those are that is a persister cell type of they shut down their metabolic activity, and that is why they survive. But in this case, this compound case, you cannot do that because their membranes are being targeted.
Okay, so there is no chance of getting it.
There is no chance of that. That will not happen in this case of compound. That's why these membrane active molecules are considered to be the next best antimicrobials.
Okay, okay. Good, good.
Thank you.
Thank you, Dr. Ghosh. Thank you, sir, for delivering such a nice lecture and providing us your valuable time. Sir, for a conclusion, I would like to request Professor Dr. Ramkrishna Thakur to give a concluding session for this webinar.
Well, thank you, Dr. Renu Singhji. And I think overall the talk and the further discussion was quite stimulating, creative, and beneficial for our student and faculty members. A lot of questions, not only from the student but also from the faculty members. I can see still you have a couple of minute time if anyone of you have any question. Dr. Naresh Sharma, do you want any question? Dr. Naresh Sharma ji? No. Dr. Shomita?
Yes, sir.
Any question?
No, sir.
Okay, great. So then I think the aim of, as you know, aim of this STP was to give an opportunity to our students to interact with the eminent scientists, professors, professionals, academicians who have actually made immense contribution to science for the societal progress or scientific development. And I'm sure throughout every session, you have got this kind of the opportunity to interact with the eminent people like today we did. So again, I take this opportunity to thank Dr. Ghosh for your time and organizing, addressing this session for the students' benefit. It will certainly motivate our students. And at the end, I would say, when you showed the picture of Berlin, the aerial view, I suppose many of our students are quite, say, interested to go to Berlin. So, Dr. Ghosh, you have to be ready to accommodate them in some of good laboratories over there or help them. So, well, again, thank you so much, Dr. Ghosh. Thank you, Dr. Renu Singhji, for your hard effort to organize this very beneficial SDP. Thank you, Dr. Sonika Sethi, Dr. Sasank Kamble, and all the faculty members of the School of Basic and Applied Sciences.
If I may say before leaving, another thing, just in case, I just want to tell all the students and faculty members and everyone present, and although we might think that probably the opportunities are better abroad and everything, but that is not necessarily true. In India also we have excellent laboratories, and we have, of course, a very big pool of intelligent minds. I'm sure some of your students are also going to fill in those gaps. So the idea should not be to go abroad only. I mean, of course, it's at some point in your life, it's necessary probably to experience different cultures, but you can do excellent science even in India. And I hope that also is there in everyone's head. Like it's not true that everything abroad is good. I mean, in India, in some of the laboratories around are excellent.
That's true. That's true. The genius mind can born anywhere, from not necessarily only in Europe or America, but anywhere in Africa, in the Middle East, in India, China. That's how we have, starting from Gautam Buddha, Gandhi, Osho, Sir Chandrasekhar Subramaniam, or as Dr. C.V. Raman, Hargobind Khurana, or from African side, we can see Nelson Mandela and many more people, societal intellectuals or scientists or lawyers or say civilized citizen. Same for the Middle East, and of course in Europe and America or Japan, there are a lot because they have a better possibility to say sign the career of a young and ignited mind to fulfill the curiosity of a student. And rightly said, Dr. Ghosh, we also do have a lot of good laboratories, but the most important thing is the genius mind. Genius mind, if they get good laboratory, say somehow support from the good people like you, certainly it would be very much beneficial for their future career and overall progress of the society or the country, or good contribution to the scientific community. It is, I suppose, it is the only scientific community which is not divided into the pieces. It is just a one scientific community. You can visit anywhere, everywhere. They can visit anywhere, everywhere, no restrictions. But for a political and some other aspect, it is quite difficult. So I believe that we all are very much fortunate that we belong to this novel profession of teaching and research, and we have an ability to somehow motivate or inspire millions of genius minds for the novel cause. With this word, once again, thank you, Dr. Ghosh, and my best regard to Germany. I had a very good impression. So I got my PhD and lot many postdoctoral assignments over there. So my best regard to all, and thank you, Dr. Ghosh. Thank you, Dr. Renu Singh and Dr. Sonika
Sir,
Sethi, all my students. Ji.
I would like to inform the speaker, Dr. Chandradeep, that Neha Tiwari ma'am is also here, if you would like to know.
Oh, okay. Hi, Neha.
Hi, Sydney. Thanks for listening. Thank you.
Okay, thank you, everyone.
Well,
Namaskar.
yeah, thank you so much. So we can close the session with these words: stay safe, stay healthy, and stay at home. That's the demand of time. Thank you so much. Thank you, Dr. Ghosh. Take
Thank
care.
you, Chandradesh. Thank you so much.
Thank you, everyone.
Thank you, Chandradeep.
Thank you, Dr.
Thank you. It is a weekend, so enjoy your
Thank
time.
you, sir.
Thank you, sir.
Thank you, sir.
Yeah,
Thank you, sir. Thank you.
great to see all of you once again. Thank you. Take care. Bye.
Thank you, sir. Bye-bye.